Hello, everybody. Good morning. Thank you all for being here today. It's a pleasure for me to be sharing this first session in cancer and inflammation on this third workshop on inflammation. And it will be an honor for us to listen to Alexandra Kiemer. And she's a pharmacist from the University of Munich, where she got her PhD also. She then had a postdoctoral training at University of Munich, and then she trained in Japan, at University of Florence, in Australia on microscopy and microanalysis in Sydney, and the Department of Immunology at Scripps. She's the head of pharmaceutical biology group at Saarlandes University from 2005 to today. And she's an expert on macrophages and macrophage polarization in cancer and metabolism. And she's going to talk to us about metabolic alterations in tumor-associated macrophages. So good morning, Alexandra, and thank you for being here with us today and accepting our invitation. So the word is yours.
Thank you very much for this very nice introduction. I'm very glad to be with you, although it's only online, but still. As already introduced, I will talk to you about today about macrophages, metabolic alterations in tumor-associated macrophages. So I will start with a short introduction into some general principles of the tumor microenvironment, and then present our data on how we characterized human tumor-associated macrophages and how we developed a new in vitro model, and then show you an example of how we modulated tumor-associated macrophages with a pharmacological compound. So I guess all of you know that the tumor itself doesn't only consist of tumor cells, but there are really different cell types also present in the tumor, and these are, for example, fibroblasts, but also a set of immune cells. And these immune cells can be recruited to the tumor from the blood vessels. This is also true for macrophages, which can differentiate from monocytes derived from the blood vessels, but they can also, macrophages proliferate within tissues. And these tumor-associated macrophages are supposed to have a role in the cancer progression, so they can really support the tumor, and this is why they might represent an attractive therapeutic target. So if you think about macrophages, this is really a cell type which is an extremely versatile cell type. It can polarize into very different functional cell types depending on where the macrophage can be found in the organism, in which tissue, but also at which state of an infection, of an inflammatory process. And so there are really lots and lots of different phenotypes of macrophages, and the two extremes phenotypes are typically denoted as M1 and M2 macrophages. M1 macrophages are really the typical inflammatory macrophage phenotype. So if you think about an acute bacterial infection, this macrophage phagocytosis produces microbicidal compounds and so on. And such an aggressive macrophage is also suggested to be able to fight against cancer, to have tumor suppressive functions, and also to interact with other components of the immune system, to be immunostimulatory, so to help fight infections, but also, of course, help fight tumors by interacting with other parts of the immune system. On the other hand, of this extreme polarization spectrum of macrophages are M2 macrophages, which are typically designated as being wound-healing macrophages. So they can promote tissue repair, they can promote angiogenesis. Both obviously is nice to have if they have a wound to be repaired, but these macrophages can also promote tumor progression. They can suppress the immune system, so also again here inhibiting other types of the immune system to fight against cancer. So as already mentioned, these tumor-associated macrophages can either differentiate from monocytes, which come to the tumor from the blood vessels. They can also proliferate from tissue-resident macrophages, and so the real polarization, so for the tumor to really become tumor-promoting, this is facilitated, first of all, of course, also by the tumor cells themselves. They can release factors, growth factors, cytokines, whatever, extracellular vesicles, which can promote the polarization of these macrophages towards a tumor-promoting phenotype, but also stromal cells can do that. Then hypoxic conditions, also dying cells. If you think about a tumor, typically you think about lots of proliferating cells, which is of course true, but there are also lots of dying cells, and tumors are sometimes designated as wounds that never heal. And so all these different aspects play together in shaping this tumor-associated macrophage phenotype, which can then support metastasis, the invasion of tumor cells from other sites, from the primary tumor to distant sites, to promote angiogenesis, to promote oxygen and nutrient supply of the tumor. So typically tumor-associated macrophages are suggested to be immunosuppressive, to be similar to wound-healing macrophages, to being similar to this M2 phenotype. However, most of the data that we have, the knowledge we have or think to have on tumor-associated macrophages is derived from mouse models. And so if people try to then model tumor-associated macrophages in vitro, they use monocyte-derived macrophages obtained from blood monocytes and then simply polarize them towards an M2 type macrophage via either using IL-4 or IL-10. We asked the question, how representative are such M2 macrophages really for tumor-associated macrophages, which can be found in tissues? And so this is why we undertook efforts to really characterize human tumor-associated macrophages from tumor tissue. So we focus on lung cancer, and we isolated these tumor-associated macrophages from cancer tissue, and in comparison with the resident macrophages of the liver, the alveolar macrophages, and we did an RNA sequencing, bulk RNA sequencing with a consecutive bioinformatic analysis. So we really wanted to know, how do human macrophages look like, which are really taken from the real patient ex vivo. And so we first did a global analysis. These were 2 patients. All the sequencing was done in 3 replicates. And I think what this figure nicely shows, this principal component analysis, that the macrophages from the healthy tissue, the alveolar macrophages, are here on the right side of these graphs, and the tumor-associated on the left graph. So the really clear distinction between the 2 cell types, whether the fact that the macrophages which were derived from a male patient, whether this is really a sex gender specific effect, this is something we just cannot tell from these 3 samples that we have, but definitely a clear distinction between these 2 macrophage phenotypes obtained from healthy and tumor tissue. So we then had those data systematically analyzed by the bioinformaticians, and so they sorted these differentially expressed genes which are upregulated, the expression in the tumor-associated macrophages, regarding their gene ontology, and we found lots of GO terms regarding to cell signaling, cell communication, and so on, which was not too surprising, right? Because I mean, we do have these tissues in the tumor, and they these cells and they interact with other cell types. And when you now look at this volcano plot, you see that here this cloud of genes represents those genes which are upregulated in the tumor macrophages, and those on the left side represent those which are downregulated. And when we just looked at some individual genes which have been suggested in the literature to be upregulated in tumor-associated macrophages. We found a lot of them. Here are just a few examples of genes involved in cell adhesion and migration. So there was not too much of a surprise. It was nice to see, well, lots of what is known is something we can also find in our system. But then when we looked at the downregulated genes, we were rather surprised to see that the vast majority of these genes downregulated in these tumor-associated macrophages were associated to metabolism, lipids, cholesterol, so really the vast majority of them. And so we again looked at some specific examples which were not described in the literature yet really to be affected, to be changed in tumor-associated macrophages. And you see here that lots of genes which are important for the production of cholesterol are downregulated in tumor-associated macrophages. While on the other hand, we found an upregulation of genes which encode for cholesterol efflux systems. So obviously it seemed that the tumor-associated macrophages try everything to keep the cholesterol levels down. And so we obviously wanted to know what is their cholesterol levels. And so we did this with a fluorometric assay. And so when we compared the cholesterol levels in tumor-associated macrophages with alveolar macrophages, we saw really significant downregulation, no matter whether we related these amounts to the cell number or to the protein levels of these cells. So this was pretty clear. We were very surprised to see this because typically there's this notion that cancers have high cholesterol levels and that these cholesterol levels promote cancer growth and progression. But in the macrophages, we really found quite the opposite. So we wanted to check whether really in these lung cancer tissues we really can reproduce what has been seen in the literature, these elevated cholesterol and lipid levels. So again, in this case, we did not isolate cells, but we took the whole tissue and analyzed the cancer tumor tissue and the normal lung tissue. Again, this was done by collaborators, which are experts in lipidomic analysis, and this was then also bioinformatically analyzed. But again, we can see in this case, we had many more analyses which we could do because for the lipidomic analysis, we just need less amount than if we want to isolate the macrophages. But what you can see here is that, again, the normal tissue and the tumor tissue can be pretty nicely separated by these principal component analysis. And when we just summed up all lipids which we found and normalized this to the weight of the respective tissue sample, you really see that we have about double the amount of lipids, all lipids taken together in the tumors compared to the normal lung tissue. And then we had all the detailed lipidomic analysis. So we, of course, looked into the cholesterol levels. The nice thing is that here we could also distinguish between cholesterol esters and free cholesterol. And you can see that in the tumor tissue, we see a strong elevation of cholesterol esters and free cholesterol if we just normalize it to the total. What if we relate it to the only within the lipids, we only see an elevation of these cholesterol esters, but definitely strongly elevated levels of cholesterol here. So I mentioned before that many people use these in vitro models to model tumor-associated macrophages, polarizing them towards M2. And so that's what we also did, compared them to non-polarized macrophages and also to these inflammatory M1 macrophages. And when we measured cholesterol here, we saw clearly that neither the IL-4 nor the IL-10 polarized M2 macrophages show a decrease in cholesterol levels. So they, in that respect, definitely do not recapitulate the term phenotype. We did see a tendency of reduced cholesterol in these M1 macrophages. It wasn't statistically significant, but definitely not reduced cholesterol levels in these frequently used M2 models, which are thought to represent tumor-associated macrophages. So we felt, well, measuring cholesterol levels is not really a detailed measure to characterize macrophages in vitro model. So we wanted to do a more comprehensive analysis and to compare these frequently used models to maybe a better model of tumor-associated macrophages. We included an additional polarization phenotype of macrophages by cultivating macrophages in the presence of tumor cell conditioned medium, which means we used a lung cancer cell line which was cultivated and the supernatant after 2 days of cultivation was collected, then sterile filtered and given onto macrophages, and is a first indication that this cell type might more represent a tumor-associated macrophage-like cell model was seen by this reduced cholesterol content in this cell model. But we really wanted to do a more detailed analysis and basically did the same as we did with the ex vitro macrophages. We did this in vitro polarization by either using these non-polarized differentiated macrophages, doing these 2 different M2 polarizations and 1 polarization, but also include this TAM-like polarization by cultivating the cells in the tumor cell conditioned medium, again, RNA-seq and bioinformatic analysis. Before I show you these data, just another brief characterization we did. We analyzed typical surface markers of these macrophages, and we saw that these M2 macrophages do exhibit stronger surface markers of M2 macrophages, what you would expect, that the M1 macrophages show higher abundance of surface markers typical for M1 macrophages. And what you can, I think, also see that these TAM-like cells cultivated in the presence of tumor cell conditioned medium, that they do show, regarding the surface markers, quite a strong similarity to this M2 cells. So definitely there is some similarity. When we then looked into gene expression comparisons, and again, extensive bioinformatic analysis were done, like clusters of differentially expressed genes, we saw that here you see the ex vivo macrophages. So the first lanes are the healthy tissue macrophages, the tumor tissue macrophages, and then here the 5 different types of in vitro polarized macrophages. And I think you can appreciate here that these cells, macrophages cultivated in the presence of a tumor cell conditioned medium are most similar to the real ex vivo macrophages, but the M2 macrophages are not so much so. And when we really did the global comparison of all the upregulated genes and the downregulated genes, we saw that there's quite a bit of an overlap of genes differentially overexpressed or underexpressed in those TAM-like cells, this in vitro model of macrophages polarized in the presence of cancer cell supernatant with the real ex vivo tumor-associated macrophages. Since we found this cholesterol metabolism genes as being very characteristic for the ex vivo macrophages, we also specifically looked into these genes. And here again, you see in this in vitro model, we see this downregulation of all those genes important for cholesterol biosynthesis, metabolism, uptake, and an upregulation of this, at least one of these efflux transporters. And this is the figure I had shown you before, and I guess you can appreciate that there's quite a bit of similarity between this really simple in vitro model compared to these ex vivo TAMs. So the question was, of course, well, is it just that they have different cholesterol levels? So who cares? Does it really have any impact on the phenotype of these cells? Cholesterol is known to affect membrane fluidity, permeability, trafficking, cell signaling, lipid protein sorting, so lots of functions in the cell. And so we only started to characterize the role of cholesterol depletion in affecting the phenotype. And so we depleted cholesterol from M0 macrophages by using the cyclodextrin, which has been known for many years to be able to just deplete cholesterol from cells. And we did the opposite. We inhibited one of these efflux inhibitors, which we had seen to be upregulated. And then we did not do yet a global gene expression analysis. We just looked at some individual genes which had been shown to be differently expressed in the TAM cells. And here you see we were successful in reducing cholesterol from these cells, and the reduction of cholesterol induced genes that we had also seen to be elevated in tumor-associated macrophages. In contrast, when we use this efflux inhibitor, this compound, here you see normal TAM-like macrophages have reduced cholesterol levels. When we inhibit the efflux inhibitor, efflux transporter, then we have higher cholesterol levels, and these higher cholesterol levels can then antagonize the gene expression changes that we see in these TAM-like cells. So to summarize this part of my talk, we could clearly show strong differences in these ex vivo primary macrophages, and the most surprising, most distinct differences were in the lipid cholesterol metabolism. And we could show that obviously the cholesterol levels in macrophages is really the opposite from the cholesterol levels in the tissue. The reduced cholesterol content contributes to the TAM-like phenotype, and that we could reproduce much of what we've seen in these ex vivo macrophages in these TAM-like in vitro model, which we are now in the present of further characterizing. Which brings me now to the last part of my talk, an example of how we were able to modulate tumor-associated macrophages pharmacologically, and the compound we used is a rather novel compound. It's a natural compound derived from a bacterium, Streptomyces strain. It's a ribosomally synthesized and post-translationally modified peptide. This is the structure of the compound. And it had been suggested to be an anti-tumor compound. And so we first looked into the actions of this compound on tumor cells, but obviously also then on macrophages. And when we did a typical cell viability assay, MTT assay, and treated the cells for 48 hours, 5 different cancer cell lines, we saw that the compound could reduce the viability of all these cells. And so actually this assay had been used earlier, and so it was said, oh, this is a cytotoxic compound, this is why it's an anti-tumor compound. However, if you look a bit closer what this MTT assay really is, it's denoted as a viability assay, but actually metabolic activities are assessed by this assay, but you cannot really conclude if you have an effect in MTT assay whether the cells really die or whether it's just an attenuated proliferation, or, well, it's just a readout parameter for metabolic activity. So we were really wanting to see, are the cells dying or is something else going on? And so we did a microscopy-based analysis in which we could stain apoptotic cells. They become green over time, and the necrotic cells in the membrane permeability stain, they become red, and this can be quantified over time. And what you can nicely see that you get this curve in these high concentration treated cells, and but not in these lower concentration, while if we look into proliferation, you see that obviously this cytotoxic concentration, these high concentration, obviously we do not have any proliferation there, but even in concentrations which are not cytotoxic at all, we see a distinct reduction in the proliferation of the cells. And so we did a comparison of the different IC50 values regarding apoptotic cell death, regarding necrotic cell death, regarding proliferation, and also then compared it to the values for the MTT assay. And what became very clear is that the compound is only toxic at very high concentrations, which are about, well, let's say 5 to 10 folds higher than the concentrations which are required to inhibit the proliferation, which by itself, of course, would be a nice anti-tumor principle. So obviously the compounds seem to affect metabolism, and so I will not really explain all the details of all these curves. So what we did is that we measured the metabolism of these cells in a seahorse analyzer, which allows to really quantify the different aspects of cellular metabolism. This is done by injecting different compounds into the system and measure the extracellular acidification rate or the oxygen consumption rate. And this is done by injecting different things, either injecting glucose to get the system more or less started, or to inject oligomycin, an inhibitor of ATP synthase in the mitochondria, and then to inhibit hexokinase by the glucose. And what we could see is that the thioholgamide, the compound, reduced the responsiveness towards oligomycin, and together with the observation that the scheme that we obtained by replacing oligomycin with thio-A, that the profile was very, very similar of the two compounds, which led us to suggest that the thio-A really inhibits the ATP synthase. And this was further supported by the fact that a very similar compound, the pre-thioviramid, did exactly the same thing. This was published by a different group. And so we think that this compound really inhibits ATP synthase, and by that affects the tumor cell metabolism. And this means that the cells can proliferate worse. We have seen this in the normal 2D system, as I've shown you, but we also see this in the 3D system. So if we generate tumor spheroids, we also see, so here you see the spheroids grow over time, but when we treat these spheroids with thioholgamide, we do not see this proliferation to the same extent. We also see reduced tumor cell migration, which is an important process in metastasis, that the tumor cells really have to move towards the new site where they want to metastasize. Again, here we did a scratch assay, and when normally over time this scratch is grown through by the cells, but this is inhibited by the compound. So is this also relevant in vivo? Yes, we did respective investigations in zebrafish embryos, injected fluorescently labeled cancer cells. And over time, you see here that there's here more fluorescence, more tumor growth after 3 days in these zebrafish embryos, whereas there is hardly any tumor growth when we treat these cancer cells in these zebrafish with this compound. So thio-A exhibits a potent anti-tumor activity by modulating tumor cell metabolism. But I promised you that, of course, we also wanted to look into its effect on macrophages. Again, we did this polarization scheme, non-polarized macrophages and the different polarizations. And it's important to know that the M1 macrophages is thought to strongly depend on glycolysis as a major energy source, while the M2, so the tumor-associated macrophage-like, is suggested to strongly depend on oxidative phosphorylation. And this, of course, this process, this ATP synthase, we saw to be affected in the tumor cells. And obviously we did the same metabolic analysis than in macrophages, in all differently polarized macrophages. And in general, we could see a clear inhibition of this oxidase-dependent ATP production, which is of special importance for this tumor-associated macrophage phenotype, which then can also be quantified as spare respiratory capacity, where the effect were most strong here in these IL-10 and TAM-like macrophages. So what does this mean for the functionality of the macrophage? You can already, if you look at macrophages, see how they might behave or not behave. And so what we could clearly see is that the compound affects the morphology of the cell. So this is typically always related to a functional effect also. And we saw that the compound strongly affects gene expression of factors which are known to be involved in tumor promotion. We also saw an effect on surface markers, M2-like polarization markers, and we also saw a distinct effect on the activity of the cells to phagocytose. So THIO-A is a metabolic regulator not only of tumor cells but also of macrophages, and it might therefore play a pivotal role in orchestrating different hallmarks of cancer, both in cancer-sized cells and in macrophages. And so to sum up, metabolic alterations really represent a central feature of tumor-associated macrophages, and therefore might represent an interesting therapeutic target, not only for tumor cells, but also for tumor-associated macrophages. And the involved pathways are not fully understood, but they really desire detailed mechanistic analysis, which we try to contribute a bit to. I would like to acknowledge, first of all, of course, the group members who did lots of the work, but also the collaborators were essential for the sequencing, for the bioinformatic analysis, for the lipidomic analysis, for the investigations in the zebrafish embryos and so on. So really thanks a lot to them. And then I think these are the group photos that most of you know nowadays from the video conferences, but these were former times, and we soon hope to be able to go back also to doing some trips and so on. And thank you for your attention, and I'm happy to take questions.
So thank you very much, Alexandra, for this very intriguing work, right? Because what is that a tumor, the high cholesterol content tumor cells do to the macrophages so that they play, I mean, in terms of cholesterol, an opposite role, imagining that high cholesterol content on the extracellular media would increase the cholesterol on the macrophages, right?
So
do you have any thoughts on that?
The egg, right? Or is this typically totally unrelated? So this is really something we cannot tell yet. Yeah.
Okay. So I have a question from the audience. So I'm very sorry that I sort of skipped the line. So Lucas Bolini, he's asking, can you read that? Does THIO-A affect alveolar macrophages differentially than monocyte-derived tumor-infiltrating macrophages? Could you compare these using PBMCs and iPSC-derived alveolar macrophages from the same donor? That's interesting.
Yeah. Very interesting, definitely. So we have some logistic limitations, I have to tell, so that, I mean, our work really taking those macrophages into culture, this really requires fresh tissue directly from the surgery being transported to our lab and so on. And so we're very happy that this works. Unfortunately, we are not able to get blood cells from the same or other types of material from the same donors. It would definitely be interesting. And the point, of course, is we would like to do much more with these primary cells, but the number is very limited, especially, of course, the number of the tumor-associated macrophages, because we need a rather large piece of tumor to be able to isolate macrophages from those at all, but we definitely plan to do more on our in vitro model, the TAM-like model, but yeah, definitely an interesting question.
Okay, so thank you very much. Unfortunately, we are running out of time. but thank you for being here with us and sharing your data. And so now I'm gonna follow inviting Robson Monteiro from the Instituto de Bioquímica to chair the next two presentations. So I think Robson, welcome.
Okay, thank you, Adriana. So let's go to the second part of this session. I'd like to thank the organizers for inviting me to coordinate this. And our second speaker of today will be Dr. Guido Lenz. Dr. Guido held the degree in chemistry from the Federal University of Santa Catarina in 1993, master's and PhD degrees in biochemistry from the Federal University of Rio Grande do Sul in 1996 and 2000, respectively. And afterwards, he was a postdoctoral fellow at Harvard University from 2001 to 2004, working in molecular and cell biology. And Dr. Guido is currently a full professor at the Department of Biophysics at the Federal University of Rio Grande do Sul and director of the Center for Biotechnology at the same university. So it's a great pleasure to invite him to talk to us today. And Dr. Guido, please be our guest. Thank you so much.
Thanks, Robson and the organizers for inviting me. It's a great pleasure to be here in this wonderful event. So today I'm going to talk about the generation of phenotypic heterogeneity in normal and cancer cells. Basically, in cell biology, we think of cell divisions and of the formation of clones as an event that produces similar cells, at least genetically similar cells. However, we more and more are seeing that even with one mitosis, but especially in the formation of colonies, we can see the generation of quite a bit of heterogeneity. Just to put this in perspective, since we are talking about cancer biology, this is at the end of all the rest of the heterogeneity that happened in cancer. As you probably know, cancers come from different tissues. Each patient has a different cancer, and different clones or even single cells have different genetic heterogeneity and therefore also phenotypic heterogeneity. I will talk today about only this square here, which is the heterogeneity generated only by phenotypic changes, not linked to genetic alterations. So we started with a very simple hypothesis. The null hypothesis is that sister cells are equal, and the alternative hypothesis is that sister cells are different. We applied this also to colonies. Either colonies are, all cells in the colonies are the same, or the colonies at the other extreme, they have cells that are as random or as heterogeneous as cells that would be randomly sampled from the same population, a population of large cells. We looked at some phenotypes we could measure in live cells, such as key signaling pathways, migration, response to therapy, and proliferation and death. Actually, we looked at fitness, which is the number of live descendants at a given time. So let's start by MAP kinase signaling. You probably know this signaling pathway. It's very important in cancer. Lots of components of the signaling pathways are changed, are mutated in cancers or misfunctioning in cancers. And we used cell line stable expressing a gene called ERK-KTR, which basically infers the state of ERG activity based on the ratio of nuclear to cytoplasmic fluorescence. And we measured the ERG activity in sister cells 6 hours after mitosis. And we observed that, yes, these sister cells are different from what would be considered our null hypothesis, but they are also different from randomly assembled pairs of cells from the same whole population of cells, meaning that some similarity is inherited in these sister cells. But when we moved to groups of 4 cells, then we didn't find any significant difference between the ERG activity of 4 cells randomly assembled from the groups or 4 cells that are from the colony. Obviously, these 4 cells, they are different from our null hypothesis. A similar result was obtained also with migration, with sister cells being different, obviously, from the null hypothesis, and cousin cells no longer being different from cousin cells randomly assembled in the population. We looked also at cell cycle desynchronization. To do this, we used the FIOCRUZ system, which stains cells, which actually expresses fluorescent proteins, red fluorescent proteins at G1 and green fluorescent protein at S, G2, and M. And we also stained the endogenous loci of Ka67 with GFP. Actually, we marked the endogenous loci of Ka67 with GFP, which, as you probably know, this Ka67 is related, its expression is related to the progression of the cell cycle. And then we looked at the size of the colony. And here's the first time that this type of graph appears, in which the x-axis is the log2 of cells in the colony. We use log2 because the number here represents the number of generations that are required to produce a colony of this size. And here we are showing the difference between the stage of the cell cycle of the leading and the lagging cell, of the cells that are most different. And what we saw is that, and here we have our null hypothesis and our alternative hypothesis, is that with the increase in colony size, the cells gradually become desynchronized. And here we can have a IC50-like number, which we call cell variance 50%, which is 3.5 generations for Ka67 and 3.8 generations for FIOCRUZ. Meaning that with about 4 generations, the cells have half of the desynchronization, and about 5 generations, the cells are as desynchronized as cells obtained randomly from the population. But in cancer, obviously, the most important measure is cell fitness, because obviously only live cells produce cancer and generate the risk that cancer represents to the patient. So we counted the number of descendants over time of several pairs of sister cells. And we compared these sisters with non-sisters. And as you can see here, yes, the sisters have differences, but they are not as large as randomly assembled pairs, meaning again that some inheritance happens in the first division in terms of cell fitness. So summarizing this first part, if we look at ERG signaling, migration speed, cell fitness, we checked other features too. This one division is sufficient to already add significant heterogeneity to these features. But in these cases, you required at least 2 divisions to not being able to differentiate colonies from randomly assembled groups of 4 cells. And on the cell cycle progression, you need about 4 generations to reach 50% of the synchronization. So if colonies desynchronize, if they the cells in the colonies become gradually different, we would expect that the colonies itself become more similar. To test this, we created this method, which we called DynaFit, short for Dynamic Self-Fitness Analysis, which basically is considered, which is based on the following rationale: that in single cells, you have a large difference among these cells. If these cells conserve their characteristics, here, for example, their growth rate, you will continue to see the same large difference among colonies. So this is our null hypothesis. But if a cell, say, with a large fitness creates a colony which has both cells with large and small fitness, and the same happens to the cell that originally had a small fitness, you will have a large difference among individual cells, but a small difference among colonies. So we organize this in a colony variance plot, in which in the y-axis we have the log2 of the variance in growth rate, and in the x-axis we have the log2 of the group size. So if it's inherited, if fitness or any other characteristic is inherited, we have a straight line that crosses the y-axis at the variance of the individual cells. And this -1 inclinated curve is formed by the randomly assembled groups, and we hope to see our data in between these two. So since DynaFit is focused on growth rate, we can say that we are measuring or inferring the dynamics of self-fitness. So we do this in the lab by growing colonies, obviously at small density, so that we know that colonies come from single cells. And then we image them at different times. We group the growth rate of the colonies with a similar size, and this gives us a given point in our colony variance plot. We produced two tools that are available at our website to analyze this data using Python that bootstraps this data and a statistical model that fits this data into a nice graph. So what did we observe? So when we checked the dynamic in fitness of a glioblastoma cell line called U251, we observed that, as quite expected, that the colonies, the variance of the colonies laid in between what we consider our H0 and our alternative hypothesis. So meaning that, and the values was around 40% of the dynamic in fitness that can be obtained gradually was obtained with the increase in the colony size. Surprisingly, other cell types, and especially primary fibroblasts, and even primary fibroblast cell line and primary fibroblasts, they also had similar characteristics, meaning that this dynamic in fitness seems to be more common than expected. At first, we expected to see this more enhanced in cancer cells, but we also saw this in two normal cells. So summary 2, colonies from cancer cells and normal cells become more similar, very probably because of the cells becoming, individual cells becoming more different. And we have around 40% of the expected variation seen in randomly assembled cells after 3 to 4 generations. And this is a common feature of cells apparently. So the next question was if this changes with therapy. So you are probably familiar with the classical dose response curve, in which you have the dose here and the response here and a nice curve, and you have less potent and more potent drugs on this graph. In our colony variance plot, we have here the colony size, here the log2 of the variance, and if the cells move upwards, the cells are more dynamic. If they move downwards, less dynamic, and they move downwards, they are more dynamic. So what happens if we treated these glioma cells with temozolomide? Temozolomide is the standard treatment for gliomas after surgery and radiotherapy. And we used a concentration that can be found in the blood of patients and that kills a part of these tumor cells over 5 days. So what we observed, actually here the treatment was 3 days, but the killing goes on after this. So what we observed is that there is a drastic decrease in the variance of growth rate with these cells with the increase of colony size. And this is even higher than what we observed in the untreated. Actually, if we compare the treated with the untreated, we see that the variability at the beginning for the single cells in the treatment condition is higher, which can be understood since cell death increases variability of the population. At the first 1 or 2 generations, we still see quite high variability, which is higher than the untreated, which can be seen that even small colonies, you will have the complete elimination of colonies due to the inheritance of the phenotype of these colonies. But then if we move to 2 to the 4th to 2 to the 6th cells, colonies of 16 to 64 cells, we can see that there is a drastic reduction in variability, meaning that the colonies, they almost behave as if they were randomly assembled from the whole population, meaning that the capability to survive or not survive to this dose of temozolomide was basically not inherited up to this point. Why is this? Well, we checked for some characteristics. This is an indicator 53BP1 binds to double-strand breaks. So it is an indicator of DNA damage. And we imaged the colonies of the cells and counted the number of foci these cells have, indicating the number of double-strand breaks that these cells had at this time. So there were no difference between the number of foci. There were no correlation between the number of foci and the colony, the number of cells in the colony, meaning the absolute degree of damage that occurs during the treatment, which is this CS2, and after treatment with temozolomide, was not correlated with the size of the colony. But if we analyzed the variation of the damage that occurred in the colony, then we see a clear correlation during temozolomide and especially after temozolomide with the number of cells in the colony. Meaning, the higher the number of cells in the colony, the larger is the variability that we see in DNA damage. We also looked at a nuclear area, which is a proxy of senescence induction. And we found that the number of the nuclear area increases with the period of the treatment. This is prior to treatment, during treatment, and after treatment. And we see that the variability of the nuclear size also increases after treatment. Looking at fitness per se, which you already saw the experiment, we counted the number of descendants of sister cells, but this time in the presence of temozolomide, we saw that sister cells, surprisingly, are not different from randomly assembled cells when treated with temozolomide. So summarizing part 3, the bigger the colonies, the more similar they become in response to chemotherapy. The difference of this dynamic depends on the cell line and depends on the treatment. I didn't show you that treating with cisplatin, for example, is much less dynamic in terms of the response to cisplatin. Another glioma cell line, A172, for example, is less dynamic than the U251, which I showed you. This variability is partially mediated by DNA damage and senescence induction, but for sure, a complex response to a chemotherapeutic agent certainly has several heterogeneities that can impact the probability of survival. The next question is if this can be modulated. So we tested a bunch of epigenetic modulators, and the data I'm going to show you, which obviously is the data that had the biggest impact, was the combination of SAHA, which is an inhibitor of histone deacetylase and increases therefore the acetylated content of histone 3 at lysine 9, and 5-azacitidine, which is an inhibitor of DNA methyltransferases. These are two broad inhibitors. They are not specific. The more specific inhibitors did not produce clear results as these broad inhibitors. So the way we did the experiment was we plated the cells. The next day we started with the treatment with the epigenetic modulator, and between time colony size 1 and 2 during 3 days, we add temozolomide in the presence of the epigenetic modulators. As you can see here, the modulator that has had the most effect on the fitness of the cell was the combination of azacitidine and temozolomide. The combination had a significant effect at growth rate 2, but no significant effect at growth rate 3. So it accelerated the death, but it did not eventually lead to more cell death after the completion of the experiment. But when we looked at our colony variance plot, we saw that the combination, these two agents, SAHA and azacitidine, actually maintained the variability of the colonies even up to colony size to the 4th. So this is in clear difference with the reduction that we observed only with temozolomide, which is shown here in green. When we looked at the single treatments, we did not see this effect of increase in variability, suggesting that you need to block the two processes in terms to maintain, to keep the sensitivity of the cells to this treatment with temozolomide. So here also, we can already see that the drug, the combination that induced the highest reduction in the number of cells was not the treatment that increased the colony variance plot, increased the variance, therefore maintained the phenotype similar to the phenotype observed by the individual cells. So the big questions we asked is, why is cell biology dynamic, or why is cancer cell biology dynamic? And is there a fitness advantage to have a dynamic growth rate? So to try to answer this, we used a Python model to make all cells divide at the average division time, or distribute the division time over the distribution we observe in real cancer cells. So this is the colony variance plot of a primary glioblastoma, which we isolated from a patient. And we used the values of the variance observed of the single cells and of the colonies here at the middle and at the end. And then we asked, what did the model show us in terms of the number of cells produced? And surprisingly, if the model uses the average division time all the time, we have more cells than if the model distributes the division time over this distribution based on the variability we observed, meaning that dynamic in fitness is not a fitness advantage per se, but it's producing a dynamic state, which I will discuss a bit later. And confirming this, we did not see a correlation between the growth rate and the dynafit status, meaning how much this treatment or this cell or this condition affected the reduction in the colony variance plot in relation to the growth rate. So we reasoned, and we submitted this in a review that is just being reviewed at Cancer Research, that colonies actually, they occupy a quite large region of the phenome, which does not give them, these cancer cells form colonies that occupy this large region in the phenome, which does not give them a fitness advantage per se. But we reason that the fact that they are continuously occupying different regions of the phenome allows them to adapt to all the challenges that are being thrown at them in terms of the microenvironment, in terms of treatment, or any other, or competition among the cells itself, that they can adapt better and thus survive. And therefore, we have all this dynamic in cancer cells. Proof of this is still lacking, although people have tried epigenetic modulators as therapeutics, but they did not produce the expected results, and probably it's very difficult to differentiate between the cytotoxic effect per se with the stabilization effect these drugs may have on the cells. With this, I finish thanking my wonderful group. They counted millions of cells by hand, and they responded very well to the challenge of doing quality science in Brazil with all the dire conditions we have here. And I thank the agencies, especially FIOCRUZ, which is our state agent, which financed our research. And at the site of our lab, you can find all the tools we developed over time to study, especially cancer research. Thank you so much for listening to me, and I'm open for questions.
Okay, Guido, thank you so much for your excellent talk and for sharing your data with us. I'd like to make a question, actually. And have you planned to use your cell dynamic model to evaluate co-cultures? I mean, tumor cells and another cell type typically found on the tumor microenvironment, such as the neutrophils or macrophages?
Not yet. What we did was to compare these cells in presence of higher concentration of cells. One of the really not so good aspects of our model is the low density of these cells. So we used higher density of the same cells stained with GFP, and we compared the results. And the results are quite similar. But we have not increased the complexity of our system, which, as you suggested, would be very interesting to add other cell types to see if this can affect this dynamics. And we didn't do 3D cultures. Sorry for reading Adriana's question here. This is a long dream of ours. But we actually, for 3Ds, we had to technically get one cell to grow into a 3D to be sure that really we have a clone being formed there. So we were not technically capable yet of doing dynafit in 3D cultures. My expectation always was that if these cells already behave as dynamically if they are alone, if they are interacting with other cells, which also are dynamic, probably the dynamic will be higher and not lower. But yes, we have to prove this experimentally.
Okay, thank you so much for our presentation. And I would like to move to the next speaker of today and to thank you again for sharing your data with us. Okay, so let's move to the next speaker. And it's my pleasure to invite Dr. João Viola to present part of his data today for us. Dr. João Viola graduated in medicine from the Rio de Janeiro State University in 1989, obtained a master's degree in parasite biology from the UFRJ-CTD Institute in 1994, and the PhD degree in biophysics from the Federal University of Rio de Janeiro in 1998. Dr. João Viola was a research associate at the Dana-Farber Cancer Institute and the Center for Blood Research at Harvard Medical School from 1994 to 1997. And he's now a senior researcher at the National Cancer Institute in Rio de Janeiro, and is current head of the Division of Experimental and Translational Research at the National Cancer Institute. So, Dr. João Viola, thank you so much for being with us today. Be our guest.
Thank you very much, Robson. And first of all, I would like to thank all the organizing committee for this great workshop in the symposium in inflammation. It's really a pleasure for me to share some of our results today. But we are going to talk today about the lipid droplets. And I would like to point out the importance of this organelle in several parts of the cancer hallmarks, including the microenvironment of the cancer, the inflammation, and the cancer progression. It has been shown a while ago that these cancer cells present at least 6 hallmarks, which including cell proliferation and evading of cell death. Later on, the same authors postulate some other cancer hallmarks, which I would like to point out today. That's the inflammation as tumor-promoting environment and the microenvironment of cancer cells and cancer inflammation will be very important to modulate cancer progression at this time. Also, I would like to point out either the dysregulation of cell energetics, the metabolism of the cell, cancer cell, also including as also very nice presentation from our first speaker, Alexandra, which also demonstrate the importance of the macrophage in this dysregulation of bioenergetics. Then I would like to show you first maybe once the first model for cancer progression in cancer, in colon cancer, in adenocarcinoma of colon cancer. In this type of cancer has been shown that the inflammation, it is a very important phenomenon in the initiation of the cancer, of the colon cancer. We have the APC mutation, just follow the hyper overexpression of COX-2, the cyclooxygenase 2, which produce important mediator of lipid mediator of inflammation, include prostaglandin. These generate a hyperproliferative epithelium, which progress to a cancer, adenocarcinoma cancer, a very invasive and metastatic cancer neoplasia. Then In this model, I would like to show you one not so new results from our lab when we analyze the cancer, colon cancer, adenocarcinoma in the same patient. And here you have in the same patient the normal epithelium and the tumor cells. What I would like to show you is these droplets of lipids with a large amount of these droplets when we compare with the normal epithelium. These lead us to study the lipid droplets on this disease. What is the lipid droplets? Lipid droplets, it is a dynamic organelle with has a crossroads of lipid metabolism and inflammation. It has been demonstrated the importance of these organelles in different diseases, including atherosclerosis, diabetes, host response to infection, asthma. And today, I would like to show you some results, what is important in cancer and obesity. For these, it is a regulate biogenesis, present a regulate biogenesis of these organelles, is the signaling platform for inflammatory lipid mediators, and as I show, has presented a role in disease and cancer. These lipid droplets have several names and depends of the species and also depends of the cells can be generated. As also as known as lipid body, adiposome, and others. It has been demonstrated the presence of lipid droplets in most species as T. cruzi, C. elegans, Saccharomyces cerevisiae, in fish as zebrafish, Drosophila, and also in adipose tissues and macrophages, include others. Then, lipid droplets is a cytoplasmic hydrophobic organelle present in wild, virtually in all organisms, including from prokaryotes to mammalian cells, as a cytoplasmic lipids with storage hydrophobic organelles called lipid bodies or lipid droplets. Has no bilayer membrane with a unique fat acid composition, has a neutral lipid core and variable component protein included. Then this organelles is not made only by lipids. It is includes several proteins. We have some structural proteins that are going to show you some data later, and I would like to point out the ADRP. That is a very important structural protein. However, these organelles or these lipid droplets also include cell signaling proteins. Of course, lipid metabolism, eicosanoid forming enzymes. And here I would like to show you the COX-2 including these. And also present traffic membrane proteins as small GTPase, caveolin, and others. Then this make the lipid droplets a complex organelle that can be involved in cancer development. First, I'd like to show you some number of lipid bodies or lipid droplets in cancer cells. Here we have 4 different lineages of cancer cell line, of human cancer cell line. including CACO2 cells. Compare with a non-transformed cells that is the epithelial epithelial cells from intestine. What you see, that is all the cell lines of the cancer cell lines present a high number of lipid bodies when compared with these epithelial intestinal cells, non-transformed cells. What you see here in this micrography, in this dot, black dot that is lipid droplets. Also, the cancer cells present a high expression of COX-2 and also produce more prostaglandin E. And I would like to show you here that this has been demonstrated that is the COX-2 and the production of prostaglandin is measured found in the nuclear membrane. However, we would like to see here that also the lipid droplets here are this dot labeling with RDP here in red, and we compare with COX-2, and COX-2 is inside of this organelle. And you would like to also to take a look, the prostaglandin synthase also is largely present in the lipid droplets. Also, this lipid droplets is a site for prostaglandin E2 production, where you have here the label of the prostaglandin E2 and the lipid droplets. What you see that has several dots here that directly produce prostaglandin E2. Then the major prostaglandin E2 in cancer cells was made in the lipid droplets. Also, I would like to show you, as I showed you before, that CACO2, that is a cancer colon cell, has more lipid droplets when compared with non-transformed cell. However, if we use non-steroidal anti-inflammatory drugs like aspirin, we block the lipid drop formation, we block the prostaglandin E2 production, and also we block the cell proliferation. And when we got the non-transformed cells, IX6, and transformed the cells with a well-known oncogene, HRAS mutated in the valine 12, and here is we transfect the cells with those oncogene. These cells start to overexpress COX-2, proliferate more, produce more prostaglandin E2, and also present more lipid droplets. Again, these lipid droplets produced by in cell transformation is able to block by aspirin, which block the prostaglandin E2 formation and also the cell proliferation. I would like to shift a little bit my talk for show you the relation of obesity and cancer, and also the importance of the lipid droplets in this crosstalk. Has been several evidence which demonstrate the association of high risk of obesity, of high risk of cancer in obese patients. That includes several cancer cell types, which we can point out the colorectal cancer, include others, and esophageal cancer, adenocarcinoma cancer, breast cancer, include others. Obesity now is associated with a chronic low level of inflammation, which is characterized by a normal cytokine production including some pro-inflammatory signal pathway. However, the molecular mechanism involved in the obesity-induced cancer is still well known. Then we would like to take a look the role of the lipid droplets in the some pathogenesis of cancer associated with obesity. I show here some labeling for perlipin 2. Perlipin 2 is the new name of the RDRP, that is a structural lipid droplets organelle. When we label RDRP, we can localize the lipid droplets. And here we have some samples, normal samples from human normal samples, obese Barrett cells, which is a pre-neoplastic inflammation associated with the esophageal cancer. And here we see the number of the lipid droplets in these samples. We start to see more lipid droplets in cancer, in obesity, esophageal patients, not related with cancer, which you see here. Basically, this labeling shows us more inflammatory cells where is related in this sample. Later, in either in cancer and Barrett samples, which is these two here, we can see a high number of lipid droplets based on the epithelium, even before the cell transformation. This suggests to us with some signaling pathway will be on this situation. And then we would like to show some data of leptin. Which is leptin? It's a adipocyte-derived cytokine or hormone that inhibits feeding and increase energy expenditure. The leptin level are regulated by nutritional status, and leptin also signals nutritional status in several other physiological systems. Also, leptin has some signaling in immune function, including inflammation, which is the signal of leptin. Leptin signaling to PI3 kinase and AKT, and activate mTOR. And we ask whether mTOR could mediate lipid droplets formation. For this, we got these IX cells, that is non-transformed epithelial cells, and cultivate them in presence of leptin. What you see, that leptin is able to induce lipid drop formation on those cells. And also, the lipid drop formation was blocked by rapamycin, which is a drug which blocks mTOR. This shows to us that mTOR could be central in the lipid drops formation mediated by leptin signaling. Also, leptin is able to induce cell proliferation, and this cell proliferation is blocked by using rapamycin, which block the mTOR activation. Then we can summarize here that the signaling of some cytokines come from adipocyte, which can include leptin. Also, we have some data that we don't have time to show, that's LDL, fatty acids, and also activated by infection pumps with activated toll-like receptors was able to induce lipid drops formation in several types of cells, which include cancer cells. These can signaling in the nuclear cell expression in several pathways which mediate cell proliferation. Cell proliferation, which includes the entrance in cell cycle, has been demonstrated, which is really related to metabolic switch in some cancer cells. We know that this cancer can be associated in this metabolic switch and include cell proliferation. For this, I would like to show some, in my talk, some data in relation of cell cycle mediated by lipid droplets. First, here's the epithelium cancer cells from colon cancer cells. And we analyze some electron microscopy. And by the way, we see a relation with lipid droplets with mitochondria, and also so intimate relation with lipid droplets with the nuclei. And here you have the membrane of the nuclei. We hypothesize that these lipid droplets can make signaling to cell enter in cell cycle in some way. For this, we made an in vitro model to induce cell cycle in NIH-333 cells, which is a non-transformed cell line. For this, we place them in zero starvation for 24 hours, and then enter with serum and see the progression of the cell cycle. At 0, 12, 24 hours, 36 hours, and 48 hours. You see that in 12 hours, the cells enter in S phase, progress to S phase at 24 hours, and enter in G2M phase later on. You see the markers at 12 hours. We start to see hyperphosphorylation of RB. 12 hours, we start to see cyclin A, that is a marker of the cells enter in S phase. And later on, we see the histone H3 phosphorylation that is demonstrate that the cells now are in G2 and M phase. Then I would like to show at this phase in G0, G1 phase at 12 hours, you see these lipid droplets labeling in green, very close to the nucleus. At S phase, these lipid bodies increase of numbers and disperse on the cytoplasm. Later on, we see the lipid bodies again, or lipid droplets, in close to the nuclei. We arrest the cells first in G1 phase by serum starvation, or in S phase by excess of thymidine in the medium. When we arrest the cells in S phase, we see the increased number of the lipid droplets, and also these lipid droplets are dispersed in the cytoplasm. You are able to differentiate G1, S phase, and M phase by labeling these cells with BRDU. If you have no label, these cells are in G1. When the cells enter in S phase, they captured BRDU, and then we are able to see early to late S phase. And by labeling lipid droplets, we see in G1 a very close relation of the lipid droplets with the nuclear. And then when you start and the cells enter in the S phase, we see the increased number of lipid droplets and disperse in the cytoplasm. Later, by labeling cells with histone H3, we are able to differentiate G2 to M phase. And what we see in G2, this still have later a high number of lipid droplets, and as the cells progress to the M phase, we see in the metaphase where the chromosomes are in the median of the cells, and the lipid droplets are in the pollen of the cells. And we localize the lipid droplets on this situation here. We don't know yet what this means, but looks like the lipid droplets really play a role in cell cycle. Finally, we transform NH333 cells again with HAS V12. And what we see again, when we transform the cells with this oncogene, that we have the increased number of the RDRP. The cells enter in S phase, and we see a really high number of lipid droplets associated with cell transformation. So we show here the association of the lipid droplets with the labeling of RDRP that is here. Finally, I would like to show you some data, again, in patients with adenocarcinoma of colon adenocarcinoma, labeling with RDRP or Ki67 for C cell proliferation, and in the same patients, normal sample and tumor sample. What we see in normal sample, we still see some labeling of lipid droplets, but this is much related with macrophage, with resident macrophage in the tissue. However, in tumor tissue, we're able to see a high number of lipid droplets of labeling of RDRP in proliferated epithelium, which is correlative with Ki67. And here we were able to see the border of normal cells or normal epithelium with transformer epithelium. And the lipid droplets really can show the border where you have transformed epithelium and normal epithelium. That could be a good biomarker for adenocarcinoma progression in human. Finally, we isolate lipid droplets by a gradient of saccharose. These lipid droplets was made of proteomics, and of course, the lipid droplets proteomics demonstrate high number or the structural proteins there, lipid signaling and metabolism, several enzymes that related to lipid signaling, and also was for us completely unexpected. The lipid droplets present different proteins related to cell cycle, and also nuclear proteins, including histone. And we still work on those experiments to see what is the importance of those proteins and the lipid droplet signaling to cell cycle and even to cancer progression. Finally, I would like to show you or finalize my talk, demonstrate for you the importance of the lipid droplets in tumor microenvironment. That includes the signaling on adipocyte, which one of the cytokines, leptin, and also fatty acids and other hormones to induce lipid droplets formation in cancer cells, and also the importance of the inflammation, where the lipid droplets can also directly produce PGE2, not only in inflamed cells, which are inside of the tumor, that's included tumor-associated macrophage, and also the signaling in the induction of cancer cells to proliferate. Finally, I hope that I can show you today the importance of the lipid droplets in several hallmarks of cancer. That include dysregulation of cellular energetics, tumor-promoting inflammation, cell cycle protection, between others that we're not able to show you today. Finally, I'd like to acknowledge the people related to this project. This project has been done in collaboration of Patrícia Bosa at FIOCRUZ, Instituto Oswaldo Cruz. The three parts of the talk has been a project conducted by Maria Teresa Scioli. That's the first part in colon cancer cells. André Cruz, which work with the regulation of cell cycle in NH333 cells. In Nayara Fazzorini, which worked with leptin and also was related to the proteomics analysis. And also, I would like to thank all the collaborators and the finding agents which can support our work. Thank you very much. And I finish here, and I'm happy to take any question that you may have.
Thank you very much, João, for the excellent talk and for the presentation with very interesting data in this fascinating field. I guess we won't have time for all the questions, but I'll pick up, I'll merge two questions from Josiane Neves, and she asked about the tumor-associated macrophages. If you have looked at the lipid droplet formation in TENS in vitro or in biopsies, and also the question that is the second one, considering all that cholesterol metabolic changes that we have seen in the first presentation, what to expect in TENS. So I joined both questions together.
I think that's a very important question, or those questions, Robson. First, what you are able to see that the composition of lipid droplets is not similar between like cells and cancer cells. You know, if we purify lipid droplets from macrophages, we purify from tumor infiltrating macrophage or other cells, and sometimes compare with the lipid droplets from cancer cells, we can see that composition is different. Also, the composition of lipid droplets can modify, depends on the stimulation when you stimulate the cells to form lipid droplets. If the lipid droplets come from inflammatory reaction like asthma, or come from inflammatory reaction like infection, sometimes they are different from lipid droplets come from cancer samples. And this difference mostly depends on the on the proteins that is inside of the lipid droplets. And that is most important for us to understand what's the difference of the composition of lipid droplets in cancer progression, and whether these inside proteins can signaling to cell to enter in cell cycle, control cell cycle, and also to regulate cancer progression. However, we don't have the final answer yet. We still work on those questions. And yes, first, the protein inside of lipid droplets could be different between the stimulus and between the cells. Also, the composition of lipid, include cholesterol, can also be different and be important for the metabolism of those cells.
Thank you so much, João. We have other questions, but I guess we have to move on. Thank you so much for the presentation. And I would like to invite Professor André Vale that will host the next presentation, the COVID-19 Storm Session 2.
Hello, everybody. Good morning to everyone. Actually, I will be replacing Dr. André Vale, who had some personal issues and could not come. So I'd like to start by thanking the great audience we are having to the third workshop on inflammation. It's really a pleasure to have you all here. And it's now my honor to introduce the last speaker of today, Dr. Orlando da Costa Ferreira, who will present the COVID-19 Storm Session. Dr. Orlando da Costa Ferreira graduated in medicine from the State University of Rio de Janeiro and had his PhD in biological science, biophysics, from the Federal University of Rio de Janeiro. He had his postdoctoral studies at the New York Blood Center and at the University of California in Los Angeles. He's currently an adjunct professor in the Department of Genetics at the Instituto of Biology, also at the Federal University of Rio de Janeiro. And he has experience in immunology and virology, with main research topics being immune response to arbovirus infections and epidemiology transfusion transmissible diseases. So it's really an honor to have you here, Dr. Orlando. Thank you for presenting this session.
Okay, thank you. Good morning to everyone. I would like to thank you, the organizer, for inviting me to this meeting, and especially would like to thank you, Claudia Benjamin, for her kindness and patience with me. And today I'm going to present part of the work that emerged from an open cohort of health professionals, which was established in the very beginning of the pandemic. The place of this work was the Center for Testing and Diagnosis at the Federal University of Rio de Janeiro. I would like to warn everyone that there will be no rock science in this presentation. It's more like of a public health presentation or topic to be discussed. And it also represents the first deep dive into the data that I'm presenting today. Which is most related to the humoral immune response in a group of individuals that was immunized with CoronaVac. I think this is an issue that is very important because not everybody in the world has experience in the immunization with CoronaVac. So the focus will be
Orlando, can I interrupt you for a second?
Sure.
To put it in a whole presentation? I think you did it already. Sorry.
Yes. That's all right. Okay. The focus will be to present information that will help us, that would help us to answer two questions. how often we will have to be revaccinated to become protected from an infection. And a second question is a very specific question that arose lately, and is related to coronavac and the waning immunity. For instance, how soon after infection memory B cells will produce enough antibody that could still protect us will protect us from severe disease of the current infection, the infection that is being acquired after vaccination. And all of this answer to these two questions are related to the possibility of having an assay that works as a correlate of protection, and possibly to be applied at the population level. So these are the topics that I would like to touch bases today. So, but before I start, I would like to show my recognition to this Núcleo de Enfrentamento de Estudos e Doenças Infecciosas Emergentes e Reemergentes, which is a new nucleus in the university that was formed from this group of people that put together this Center for Triaging and Diagnostic of COVID-19 at the Federal University of Rio de Janeiro. So this center that starts screening the population, the whole population of Rio de Janeiro, and the employees of the Federal University of Rio de Janeiro, was started in March 16, in the very first few days of the pandemic. And as September 28th, it assisted 23,000 people, have done PCR, 55,000 PCR, around 35,000 serological tests, and immunized more than 6,000 people, and among many other activities. So this is the Center for Testing and Diagnosis today, and on the right side is the future of the nucleus. I'm just showing you an image of the future. This is the nadir, as I said, is a new nucleus of the university. It will be held in this building that is located at the old where the BioRio was previous is located nowadays. So this is the future of the Center for Testing and Diagnosis that started in March last year. So what are the issues to be discussed? First, as I said, is a correlate of protection. And the cohorts and the cohorts that will discuss these issues will be the cohort of infected individuals that we've been following since the beginning, and this is a dead. I must say, this is a dead cohort because everybody now has been vaccinated or immunized, and then this gives rise to the second cohort, and these immunized individuals, and of course, individuals that were previously infected got immunized and constituted the third cohort. And the fourth cohort represents the breakthrough infection, infection that appeared in individuals already immunized, especially for this group of cohorts that I'm presenting. I'm talking about immunization with the coronavac vaccine, the Sinovac vaccine. So what kind of data we'll be presenting? We will be presenting data from sequential specimens that has been collected from individuals participating in the study, and we analyze the presence of the three types of immunoglobulins and the neutralizing antibody that arose from these individuals. Altogether, in this analysis, during this time, we had about 18,000 participants that generated over 35,000 blood specimens. And of the total of these participants, single bleed were obtained from 9,000, and the remaining 8,000 and something individuals donated between 2 and 23 specimens. The data that I will present today will analyze 1,236 individuals and samples and 6,653 specimens. Okay, I'm start talking about the correlate of protection because this is a very hot issue now in the literature of vaccination. Okay, we know that that all three types of immune response are involved in the protection against COVID-19. So there were in the beginning there were many discussion about which one, the innate, the T cell mediator, or B cell mediator, would be the most important. And I think, in my opinion, all three are important. But and the voice of the world is claiming for a simple correlate of protection. So there were many papers that appeared in the literature in the last 1 or 2 months that are calling for the need of a correlate of protection for SARS-CoV-2 vaccinees. And especially this one that I'm presenting here on the right, that this is a publication that appeared 2 weeks ago in Science, and was about the correlate of protection that using the AstraZeneca vaccine as a model. And this paper clearly established the four correlate of protection based on detection of antibody against the spike, the IgG antibody against spike, IgG antibody against the RBD, which is the receptor binding domain of the spike protein, and 2 other assays that is one of them using pseudovirus, and the other the live virus or authentic virus in the format of plaque reduction neutralization test. So this, according to this publication, these 4 assays were associated with protection to asymptomatic infections after vaccination in individuals that got vaccinated. But more important, this paper describes potential levels of antibody that is associated with protection, and also titers of neutralization titer when the assay measures pseudovirus or live virus. And the parameter to establish these target values are now. I entered the site yesterday. The site is not working. I mean, I was not allowed to get into the site because I think that many people are looking for those antibodies to calibrate their assays. So they have now international units to validate every one of these 4 assays. Okay, just as a reminder, if you don't know, I mean, using humoral response or antibody assays as a correlate of protection is the most common method for measuring protection against several infectious agents. Assays is as simple as ELISA or even hemagglutination test that is used for influenza. So why is this important? Because this is important because we can apply. I mean, you no longer need to do a clinical trial with millions of people to define the efficacy of a vaccine. If you have an assay that can measure if the individual was immunized and the level that immunization that we reach is correlated with protection. So I think we can rely on the measurement of the antibody in the patient's serum the two associate with protection from symptomatic infection, at least. That's what the paper said. And here I'm showing you, I'm sure that you are aware that I'm showing you that the two of the fourth assay that the paper mentioned. One is the plaque reduction neutralization test that measures the inhibition of the penetration of the virus in viral cells and uses the authentic SARS-CoV-2 virus in a BCL3 facility. So it takes 3 to 5 days to complete the work, and it's not easy to perform. It's very time consuming and has biosafety issues. There is a simplification of this method that use a pseudovirus, which is mostly retrovirus or adenovirus that is expressing the spike protein on the surface and also put in contact with the cell as shown down here. Then you plate your, this is a monolayer of susceptible cells. In this case, it's a viral cells. And then you put the cells in contact with the virus, and the virus kills the cells and make these dots on this carpet of cells, and that's what you measure in the colonies, that is the plaque that is producing by this infection, and you then to define the titer, you add the cell, the virus, and the serum from the patient in different dilutions, and depending on what you want to measure, if you want to have 90% inhibition or 50% inhibition, you have a measure of the titer that is able to inhibit 90%, which is this case, or 50% of the infection. And this is how you measure this sort of assays, and most of them are very complicated. So we look in the literature to what would be the option for us to use a simple assay that would be able to screen lots of people, and that would be easy to do. So we encountered this Lumit assay, which is a homogeneous assay. Homogeneous assays is an assay that doesn't need a solid phase, to separate the complex, the immunocomplex that was formed during the antigen and antibody recognition. And so it's a very practical, for the reason that if you had to use a solid phase assay in which you would like to block the intercept of the RBD and the ACE2 with an antibody, we need to control the equimolar amount of both RBD and ACE. So if you put the ACE that is commonly used in the solid phase assay as a solid phase reagent, then it's difficult to control the amount of the ACE that is bound to the solid phase. And it leads to a lot variation and difficult to maintain to account for the relationship between these two amounts of the reagents that you need in the assay. So an homogeneous assay doesn't need that. You do the reaction in solution. You know exactly how much of each of the reagents you're adding, and the only thing that varies the amount of the antibody that is present in the sample. So schematically, this is a presentation. So they use an RBD that is linked to a FC receptor, a immunoglobulin FC receptor from rabbit, and the ACE that has an FC receptor from a mouse. Sorry. And then you mix the two together in the presence of the serum samples that contains antibody against the RBD. Okay, the other component of the reaction is a Lumit that is the luciferase that is divided in two pieces. and is only brought together when this antibody recognizes both the FC of the rabbit and the FC of the mouse. And they do only get together when the ACE binds to the RBD. Okay, so it's a very easy reaction, and when they got together, it emit lights, and this detected in the luminometer. So the reaction takes about 2 hours to occur, and the cutoff is you measure the percentage of the binding, and then when you have 70% of binding, inhibition of the binding, then you have identified the neutralizing antibody. And this experiment that we did to correlate the amount of correlate the percentage, the limit, and the PRNT90 with the limit, and you see that the higher the PRNT of the titer of the sample, the higher is the limit assay as well. So we use this assay along with the ELISA to detect all the immunoglobulin types to analyze the data in this cohort. I would say a few words about the incubation period, symptoms, onset, and testing. Just to mention that the incubation, it's a symptoms onset. It depends on the incubation period, and also and how the patient perceives the symptoms of the COVID-19. So it has a variation for each patient. But nevertheless, what you have for testing for diagnosis at the first few days, the most reliable assay is a PCR, and that's what you use to do to diagnose our cases in the CTD. And after 10 to 15 days, then the serology becomes the most important marker. So this is just to point out that the viral proteins that are relevant for antibody response. For this study, we're going to show you the results using the trimeric spike proteins, some data with the subunit S1 of the spike protein, and the interaction between the RBD and ACE2, which is the limit assay. So this is the first, this is an old slide, just to show you the results of 300 individuals when we set up all the assays that we use during this study. We set up the IgG anti-trimeric protein, which was a kind donation from Professor Leda Castilho from COP, who has donated the trimeric protein since the beginning of the pandemic. And pretty soon we established that between 10 and 15 days, we had all of the individuals had the diagnosis, had presented immunoglobulin in the serum. And this level of immunoglobulin, this is doesn't seem to vary along the time. Okay, this is initial data with 4 months follow-up of some individuals, and you see that the level doesn't fall. But when we use the S1 part of the spike protein, what you see is this is a similar kinetic as the trimeric protein. It's a little bit delayed in the detection, but the antibody tends to fall with time. This is the data that was generated in Dr. Andrea Valli lab using the RBD as antigen. And you see that the antibody against RBD show up a little bit later and tend to disappear. Some patients still remains with antibody. The IgA titer is also measured. We have assay for S1 and alpha 3 for the trimeric IgG. So one thing that we also was able to develop was a nucleoprotein assay for some individuals. That this represents a follow-up of one individual that was vaccinated with coronavac, and you see that vaccination for this particular individual doesn't induce antibody against the nucleoprotein, but against S protein, after the second dose, it goes up. However, if the patient gets infected after vaccination, the nucleoprotein emerges at a very high speed and similar to the one that appeared with the antibody against the trimeric protein. Okay, so here is the first cohort. The cohort 1 is of infected individuals. I'm going to show you the data of 343 individuals, totaling 1,500 samples. Okay, this figure here on the left shows the kinetic. This is individual that we've collecting sequential samples. Every line here represents an individual, and the dots are the time when the samples were collected. Here is the time from the infection or since the symptoms onset to sample collection. And what you can see is that there is no decay in the IgG level even about a year of follow-up of these patients. This is like now he just shortened the window period and to show that there is a kinetic of antibody production that between day 10 they start appearing, and between day 10 and day 15 they reach. Everybody has already IgG against a trimeric protein. Again, this is the data showing with extended the data now, and you see that there is a constant decrease in the IgG antibody against S1 along the time. And also, these are data with IgM. As expected, the IgM goes up, and after 3 to 4 months, you don't find IgM, and sometimes you find a very borderline levels of IgM. IgA also increase very rapidly, but tend to disappear after 2 or 3 months of infection. Also relevant to this presentation is the detection of the neutralizing antibody with the Lumit assay. So what this assay is detecting is the antibody that is interfering in the binding of the RBD with ACE. We can discuss later if this is the only neutralizing antibody that the individual generates, but this we can leave it for discussion. And so what you can see here is that also there is a decay in the level of neutralizing antibody, as has been suggested by the previous study with the antibody against the RBD that I showed in the previous slides. The Lumit also has a kinetic for the appearance of antibodies. Most of the antibodies will appear after day 20, but it can, for a group of patients, that it can appear at the same time as the anti-S antibody. And this is the most important. This is the data that we have from the infected cohort. As I said in the beginning, this cohort is dead because everybody now has been vaccinated, and this turns on to the second cohort. So we measure for this cohort. We measure only IgG against the trimeric spike protein and the Lumit of patients immunized, individuals immunized with coronavac. What we can see is that this is the time of first dose of coronavac, and the second dose was 28 days later, which is mid-time between these 2 points. And it reaches a peak 2 weeks after the second dose. Okay, so it's basically day 42, which is about here. And what you see after that is a continuous decay of IgG, which we have not observed with infection, and also a decay in patients in the neutralizing antibody at a very faster decay compared to the infected individuals. So this made us suspect, this is a fact now that we know, that IgG and the neutralizing antibody decay over time, and this is the alert sign that the waning immunity of this coronavac vaccine. When we look at the individual patient, you see that compared to the infected individuals, what you see is that over time, the lumen decay is lower after infection than after coronavac immunization. Okay. The decay of lumit is much less dramatic after infection than after immunization with the coronavac. So we then divided the neutralizing antibodies, or the lumit assay. I'm using the lumit and the neutralizing antibody as synonymous here. And we divided the patient that did not reach the 70% level of that would represent a presence of neutralizing antibodies, and called them non-responders. And we distinguished them from the patient that reached and passed the level of 70%. Okay, and these were considered patients that never acquired neutralizing antibody and represent 35% of our population. So 35% of the patient that was immunized with coronavac in our data, which is a small number that is here that we measure this antibody, it was not fully protected against infection. Among the ones that reached the level of the neutralizing antibody, what we saw was that we were able to draw a line, a regression line, and find the rate of the antibody decay. And you consider that if a patient had 100% of fully protected of the maximal level of detectability of this assay, then it would take them 86 days after reaching the peak to lose the antibody, to cross the 70% line. Okay, so basically this is how long individuals get protected if they reach the top level of the neutralizing antibody after being immunized with the coronavac. In other words, if you immunize with coronavac and you wait until you get the second dose, and you wait until you get your top level antibody, neutralizing antibody, then 3 months later, you're no longer protected. This is the similar graph, but using the IgG. You see that the IgG level among the responders are higher than among the non-responders. Okay, so the cohort 3 represents the individual that were infected and then were immunized with coronavac. And we have 188 individuals in such cases, and this contributed to about 1,000 samples for analysis. What we see with IgG levels, you see here that the patient got, time 0 represents the first dose of coronavac. Okay, what you see here is the individuals got infected, the raising level of IgG, but when they were immunized, this level increased for almost all individuals. Some individuals have the decreasing level of IgG, but the most majority of the individuals had rising level of antibody after immunization. The same thing happened to lumit, except that although some individuals reach the top level of the neutralizing antibodies in this assay, other group of individuals tend to lose this antibody even after having been infected and after having been vaccinated with 2 doses of coronavac. Okay, so the same. This is just the IgM assay, just to show that the IgM levels increases after infection, then it decay, and very few individuals present with high level of IgM after being vaccinated. IgA has a similar profile as IgG, but some individuals developed high levels of IgA and some others don't. But one of the things that made us think about, especially with the Lumit assay, is the probability that how far the immunization has to be from infection to have levels of protection maintained over time. So we divided our infected group in 3 parts. Individuals that was infected, that had the symptoms onset 200 days before the first dose, between 100 and 200, and less than 100 days. And what we can observe is that it seems that, in the interest of time, I have to speed up my presentation. It seems that the far apart is the infection from the immunization, the better is the neutralizing antibody response after immunization. Okay, if you see comparison, most of individuals in this group that was infected far away from the immunization, they have maintained their level of a neutralizing antibody. Some has lost, but you see in this case here that the individual that was infected like 100 days before immunization, the sustained level of the neutralizing antibody is not quite good, and most of the individuals decay their level of neutralizing antibody. So I will skip this slide because it's just to show some data. This is this is related to IgA. It's not so important. And this is the comparison of the Lumit and the AstraZeneca. I said that I was not going to talk about any other immunizer, but I just, for comparison, if you take the individual that has been pre-infected and then immunized with coronavac, and compared to the same group of individuals that was immunized with AstraZeneca, what you can see a pretty neat difference between AstraZeneca and coronavac, which most individuals in the AstraZeneca maintain their high level of neutralizing antibody, and the coronavac continually decaying their level of neutralizing antibody. So yes, there are differences between in the vaccines, and here is just to show the large data on the extended data on these 2 cohorts. This is immunization with coronavac and AstraZeneca only, just to show where this is the red arrow shows the time of second dose for coronavac and AstraZeneca, and the decay of antibody of the peak of the antibody in these 2 vaccines. And you see that the peak of the AstraZeneca comes after the second dose, and the AstraZeneca, the peak comes, we expect, we don't have the large data yet, but we expect that this high level is maintained over time after the second dose. Okay, the 4 cohort is the one that matters to us. Is if we patient immunized with coronavac, and if they get infected, what happened to them? We have 125 individuals in this group contributing to 654 samples, and these represent breakthrough infections, and this is the most important to us right now. So here we're comparing if the individuals get infected between doses and after the second doses, okay? And you see here that the individual that got infected between the first and second dose of the coronavac has a much lower level of antibody against the trimeric, the spike protein than the individual that got infected after the second dose. Okay, so if there is a breakthrough infection, what we expect of these individuals for the future. Okay, so this individual that got infected after the second dose has a better level of IgG antibody than the individual that was infected between doses. The same thing here is just showing the kinetic. The kinetic, I think we have a delayed kinetic for infection between doses, meaning that the individual tend to delay the increase of IgG in regards to the infection between doses. And if they do get infected after the second dose, you have a faster increase in IgG levels. The same we can see here in the level of neutralizing antibodies. Between doses, you see that many individuals reach the top level of the antibody, but they start losing the antibody very fast. And for those ones that got infected after the second dose, you see a maintained level of the antibody. But most important for us is, which I would like to stress in the discussion, if you have time for that, is that after the infection, how soon we developed antibody. Memory B cells will produce antibody to protect us from this current infection, from having a serious or severe disease. Okay, what we have observed is that this antibody, the neutralizing antibody, appears in after the day between day 4 and day 3 and 4. In other words, up until day 3 and 4, nothing happens. I mean, everything remains the same. But after the day 4, people start showing neutralizing antibodies at the high level, and this is exactly the same window that individual that uses remdesivir, which is an antiviral agent, anti-SARS-CoV-2 agent, and monoclonal antibodies. They benefit the best when they use those medications in the beginning of the disease. So we believe that the rapid response of the neutralizing antibody upon reinfection, it warrants that this individual may not have a serious, severe disease. So this is another data. If you further divided the infection that occurred between the two doses in those that occur before day 12 of the first dose, and the one that occurred after the day 12 of the first dose, you see a clear difference here. You see the red is the infection that occur after the day 12, and the blue lines represent individuals that got infected between day 12. Okay, so those individuals doesn't have a complete neutralizing response, and this is just a close-up view of this figure. So infection after day 12, after 12 days after of D1 shows an early and sustained and neutralizing antibody response then between day 1 and day 2. Okay, again, this is important because maintaining this and a quick response of this antibody level will help to protect this individual from having serious disease. And I guess this will be my last slide. How about the third dose? I'm going to show you an example of two individuals. One of them is this one over here. Is more. He was immunized with coronavac with D1 and D2, and about 6 to 9 months later, he received the third dose. But I would like to draw your attention to the fact that these individuals, after the second dose, got a neutralizing antibody that was falling really fast over time. And in fact, after a few weeks of the peak level, he no longer had the neutralizing antibody. This low level were maintained until the immunization with the third dose. Okay. I also mentioned that the IgG antibody has fallen so low that the assay was negative. So technically, this individual could be considered an ex-vaccinee. Okay, so he no longer had antibody against COVID-19. So, but upon the dose 3, I mean, this is not the dose 3 of the coronavac. This is a Pfizer vaccine. There are 2 points here, divided by 4 days. Nothing happened during this period. But then at day 7, the IgG, pardon me, this is these 2 dots here are separated by 4 days. Nothing happened for neutralizing antibody, nor the IgG. But at day 7, after the third dose, the IgG really high, and the neutralizing antibody is reaching the level of neutralization. It's about to be a neutralizable antibody, okay, antibody level. So this is an individual that got infected. He lost neutralizing antibody, was infected after the second dose, maintained a high level of antibody, and took the third dose, and still maintaining the third dose. No difference in the profile, but this one would benefit the most of the third dose. So we do believe that the third dose will be important for some individuals that have lost this neutralizing antibody, and maintaining this level high will help him also to be protected from severe disease. Okay. Finally, I would like to acknowledge and thank you, this spectacular group of people that help us every day to maintain the high level of research and assistance to the patient that look for diagnosis and counsel. Without them, we will not be able to reach the level of quality that we have maintained in our group. Thank you very much.
Thank you, Dr. Orlando. Unfortunately, we are with our time very short, but we have a few questions here. Maybe I'll have to reduce it to 1 or 2. So Dr. André Valli is asking if the second wave of antibodies is able to neutralize a broader range of variants.
Well, that's something that we haven't analyzed yet. But I think that every time that, especially after infection, even if you are immunized first and then you have an infection, I think that your breadth of your response is increased. That is being shown for other vaccines. So I believe that with a third dose, we will reach a level of a neutralizing antibody that will, with a breadth, a very wide breadth of recognition of variants. But this is to be tested.
And Dr. João Viola has pointed that you have very important results on humoral response. But he asks, how is the cellular immune response on those individuals? If some individuals present low levels of IgGs, but may still have cellular response, let's say CD8 T cells and protection.
Yeah. Okay. So this is a very important question, and because people there is a long-lasting debate about the importance of T cells and the humoral immune response. I would say that the consensus in the literature is that if you don't have antibody, okay, so if you don't have humoral immune response, the individual will have a serious disease. He may not die because he was protected by the cellular immune response. But if you have a humoral response, this individual would receive a much more lower burden of virus and probably will not develop a symptomatic disease. Okay, so the answer is kind of so-so, but I think that the antibody has, there is no doubt that T cell protects from disease, from serious disease, but the antibody has its role in the protection. And I should remind that a correlate of protection doesn't have to be associated with the mechanism of protection. It has to simply indicate if the individual is or is not protected from having a serious disease.
Thank you, Dr. Orlando. I'd like to thank you again for the excellent presentation and also to all the other speakers, and thank the audience for the day. I'd like you to stay for a little longer, and I'll call Dr. Robson Queiroz so that he has a special invitation to you all.
Ok. Thank you, Rosane. Eu vou falar em português agora, gente, só para poder convidá-los para um evento que será realizado que que será realizado online também daqui a algumas semanas. É o 15º Simpósio de Oncobiologia. Para aqueles que não sabem, tá? Acessem nosso site, simpósiooncobiologia.com.br. Temos uma programação com vários palestrantes na área de biologia tumoral. Então, convido a todos para olharem lá rapidamente. A recepção de trabalho já está encerrada, porém, a inscrição para assistir o evento ficará aberta até o primeiro dia de evento, que é no dia 8 de novembro. Então, convido a todos para visitarem nosso site, novamente aqui, simpósiooncobiologia.com.br, darem uma olhada lá nos palestrantes. Teremos sessão de apresentação de trabalhos, já encerrada a submissão de resumos, mas aberta aos inscritos. Ok, então, mais uma vez eu agradeço a possibilidade de fazer essa divulgação. Rosane, it's up to you.
Just to end our presentation, obrigada, Robson, obrigada a todos que estiveram até aqui agora, boa tarde a todos. E nosso próximo evento é sexta-feira.